Right, let's start. So today's speaker for the NeCEN Seminar is Cristina Paulino from University of Groningen. Cristina did her PhD in Germany with Werner Kulbrandt. Did you do 2D crystallography?
Yeah.
Yes, so I also did 2D crystallography.
Yeah,
so
the unicorns. Yeah,
yeah, the unicorns.
yeah.
And then for a PhD, and then Cristina did a postdoc in Switzerland, still doing membrane protein structural biology. And she joined Groningen last year as an assistant professor and head of the cryo-EM facility. And looking forward for your talk.
Yeah, thank you. So yeah, so I'm currently since roughly a year now established my own group, and what I still focus on. So I will today talk about this specific project which we just finalized, and the paper actually just got accepted 2 weeks ago. But just to give you a brief overview of what I'm actually interested in. So I'm a biochemist from house, became a structural biologist, always focused on membrane proteins for understanding how they actually work. And I mean, if you look at the cell, the membrane proteins, there are of course quite a lot of them. They're like classical enzymes, receptors, junctions, but these are not the ones that I'm interested in. The ones that I'm really interested in are the membrane protein transporters, so all those proteins sitting in the membrane which facilitate in one way or the other the transport of substrates or other compounds from one side to the other. And we humans like to always put things in boxes and dogmatize and classify things. So if you talk about membrane transporters, we always classify them into 3 distinct groups. So the first one would be what we call a channel. So this facilitates the passive transport of normally ions, but it could be whatever, along the electrochemical gradient. And then we have the active transporters, which then basically transport a substrate or compound against the chemical gradient. And here you then start dividing again among secondary active and primary active. This is just the source of the energy for this process. So if you have a primary active transporter, this is where you have a direct hydrolysis of ATP for this. So and I really work on or have been working on all 3 classes. For those that have been at the NVVM meeting in Groningen in May, you might have heard about this story, so the human ACT2, so it's an amino acid transporter which we solve. So this is a very classical secondary active transporter. But what I'm trying to focus more and more in my research field are actually those that do not obey to this typical nomenclature, so which challenge these conceptual boundaries. So I like to challenge dogmas and rules. So one of them is this TMM16 family, which members can act as channels or lipid scramblases, which, well, from a technical point of view, would classify as secondary active transporters. And what I'm going to talk about today is the KDP-FABC complex. We were just discussing about this terrible names. I'm sorry for this. I can't do anything about it. I did not invent the name. So in the KDP-FABC complex, really, it's actually different than the TMM16, where what we have here is actually a complex of a channel and a primary active transporter coming together, and this is actually very intriguing, and this is also the reason why I fell in love with it, and we started working on it. So from again, like if you want to classify it, you would put the KDP-FABC complex into p-type ATPases. So you probably all heard of it. So formerly they were classified in this type of like the type 4, 2, or 3. Now there's actually a new nomenclature, already since quite a while, but people don't like to stick to it. But I'm pretty sure that you all know at least 1 or 2 examples of p-type ATPases, and this would be the sodium potassium ATPase. So this is the one that in our cells can consume up to 75% of all ATP produced. So it keeps up the sodium potassium gradient, which is then used for the secondary active transporters to do their function. And you probably also heard about the calcium ATPase, also known as ZERCA. So this is the one that sits in the endoplasmatic reticulum and really tightly regulates the calcium, which then acts as a secondary messenger. So overall, p-type ATPases are known as ion pumps. So they belong to the primary active transporter. So they hydrolyze ATP to pump then an ion against its chemical gradient. But it turns out the family is actually much more complicated, because we have also flipases, so ones that transport lipids, and there's quite a lot unknown. And KDP, this complex belongs actually to the tiniest of them all, and it's only found or still found in E. coli. It doesn't spread to eukaryotes, but it's still a very striking one. And we'll come to this a little bit later. So just to give you another better overview, peptide ATPases, so what they do, I already told you, they hydrolyze ATP, and then you normally can divide the structure into a transmembrane domain and a cytosolic domain, which is composed of 3 different subunits. So one binds the nucleotide ATP, the other, the blue one would be the side where you have the residue which becomes phosphorylated during this process, and then you have the acute domain, which then comes in proximity to dephosphorylated. And what happens in this cycle, so this is, we actually know quite a lot about p-type ATPases already, and in this E1, E2 cycle, what happens is that due to the hydrolysis of ATP and dephosphorylation, so the phosphorylation, dephosphorylation of the protein, you have very large conformational changes. This is what you can see here, and they then propagate a conformational change in the transmembrane domain, which then alters its accessibility, so that you have it sometimes open to the outside, sometimes to the inside, and this is then how the ions can go from one side to the other and are actively pumped according to their function. So where does KDP-FABC now come into play? So I told you already that it belongs to a very small family, only found in prokaryotes. If you talk about the function, it belongs to the superfamily of potassium transporters, so SKT. And what is perhaps also very striking is that it's normally not expressed. It's really only expressed in emergency situations, so as a survival machine, because you actually have other SKT transporters which normally take up the potassium in the cells. But if the gradient drops so dramatically that the other transporters are no longer capable of pumping potassium inside the cell, this is really where then KDP-FABC becomes expressed. So it's an emergency protein, let me call it that way, and it's really a unique chimera. Why? I mean, if you just remember from my introductory slide, we normally have this well separated where people don't mix these boxes of a p-type ATPase, so a primary active transporter, and a channel. And in this complex, we actually have them coming together. So what we have is, we have this one subunit which is called KDPA, so now the name should become a little bit more elusive. So we have this subunit. It's a fairly small one, and it resembles pretty much what we know about the KCSA potassium channels. So it has this typical 2-helix and this P-loop, which informs the selectivity filter. What we find here is that it's all encoded. It's not a homotetramer, but it's encoded all in one subunit. So we have transmembrane helix with this motif, and this subunit has been since decades postulated to be the one that actually mediates the ion transport. So here what I depicted as the brown subunit is KDPB, so this is actually the one that belongs to the p-type ATPase, so that's also why you categorize this whole complex into p-type ATPase. If you look at it, it has actually a smaller N domain, and also the transmembrane domain is really just a core domain. It has 7 instead of 10 transmembrane helix, so it's somehow a little bit turned down. And what always has been postulated is that it presumably has no cation binding site, so this canonical binding site, which is normally the one where you have the ion during the ion pump process binding, that this is disrupted. And what was thought is KDPA is the transporting unit, and this is the energizing unit. But there are, of course, 2 other letters, if you remember it. So C and F, so these are smaller subunits which all come together. So we have KDPC, which is pretty small. So it's also just one transmembrane helix, but still has this periplasmic soluble domain to it. And KDPF, it's really just one helix in the membrane, and they are rather thought to have more of a chaperone or catalytic chaperone function. So altogether, this is 157 kilodalton, is asymmetric. So, I mean, nowadays this is not necessarily that big of a challenge for cryo-EM, but it's certainly not an easy target. So, but what makes me fall in love with it, and it's so intriguing about this system, is, as I told you, it's really a chimera between a p-type ATPase and an ion channel. It is thought to have a spatial separation of transporting an ATP hydrolysis, so you don't have it transporting via here, but only via here. Then this raises a couple of questions, if this is really how it works. So first of all, how is the energy produced here, even coupled to this subunit. So this is the first question we would need to answer. And then the other problem is, I mean, we are pumping, actively pumping potassium with a very high gradient difference from the extracellular side to the intracellular. So we cannot be working with a channel. So there has to be somehow an unidirectional gating that prevents potassium to just flow in the other direction, which is what it would of course do in a channel type subunit. So, however, these two questions are solved. What is very clear is that we have a completely new, unprecedented mechanism of action, and this is, of course, what is striking. As always, when you start something, there might be someone that is quicker than you, and, well, I would not call it that we were scooped, because I think you can always do the best out of it, so you can just, well, I took it in a very optimistic way, and I will show you also why we even benefit from it. So this was just in 2017 when I started working on, or actually already had started working on it in Zurich. There was an X-ray structure solved, which then revealed also the general architecture of it, because before that, well, it was just a couple of assumptions. It is at 2.9 angstrom resolution, and probably in an E1 nucleotide-free state, and at the selectivity filter, here they even saw a potassium bound. So what did we learn, or what did the authors learn or proposed by looking at the structure is, first of all, they identified, so also just to, so there will always be the same color code. So this is the p-type ATPase subunit, and in green is the channel subunit. So this is the most important thing you always have to keep in mind. So what they found is they were looking at the structure, and they realized, oh, there's actually a tunnel, that it's an empty space that spans from the KDPA, so from the channel-like subunit down to the p-type ATPase subunit. And what they propose is that this is a proton via tunnel connecting both subunits. So what they propose with this is that you have water, it's a water-filled cavity, which basically would sense a charge here and then signal this charge down to the former canonical binding site, and this would then induce the conformational changes and get the process rolling. This is what they suggested, and what would then happen is if the signal comes down here and the conformational changes take place, there's another very particular structural element in this whole complex, and it's what they depicted here as pink. And this pink helix still belongs to the channel subunit, so KDPA. And what they propose is, if you look at here, it's a zoom from here, is that you have the blue, this blue helices still belong to one of the subunits of the KDPB, and it's via a salt bridge connected to this helix of KDPA. So what basically they propose, and I'm just showing it here in this schematic view, is that if a potassium binds through the selectivity filter of this subunit, the proton wire tunnel will basically send the signal and induce conformational changes in here, and these conformational changes would basically then pull on this connecting coupling helix and open up the channel. So this is basically what they propose, and I mean, of course, as I told you, you have still another problem which you have to solve, how do you close it from the other side, because otherwise if you have a pore, this would never pump against the gradient. What they suggest here, if you just depict this other subunit, it's the C subunit, that this would then just change conformation and just close it like a lid. So that's basically what they proposed, but we were still intrigued, and nevertheless we already started working on it during cryo-EM. And so since they solved a so-called E1 state, we were trying to capture an E2 state. For this, we chose to stabilize it with a substrate and inhibitor mix. So these inhibitor mixes are also very well known in p-type ATPase studies, because these are analogs, ATP analogs or phosphate analogs. So this is how you can mimic and trap these proteins in different intermediate states. So we use this combination, this cocktail, because it's known to normally stabilize an E2 state. Well, at the beginning, as so often with cryo-EM work, we had some problems with sample preparation. So at the beginning, we struggled for quite some time, that we had what I always call this microaggregate. So you see sometimes also some single proteins, but we really struggled with this. But we finally also made it. So the take-home message here is really prepare your samples fresh. So I mean, this was also during the transition time when I moved to Groningen. at a certain point, I mean, we started also this collaboration then with someone in Frankfurt to join forces, because otherwise we would not make it in time. And then the deal was really, okay, we need to purify it where we freeze the samples right away, and this really made the difference. So that we then were finally able to obtain a good data set. I'm not going to go step by step into the cryo-EM workflow, but we really started with an extensive number of micrographs, and already very early on during the image processing workflow, we realized we actually have a heterogeneous data set. So we had at least 2 populations in it, and this is also what we at the end managed to separate, so into 2 distinct populations, which we call state 1 and state 2. I mean, here I can also just very briefly show the validation. So we have then one of the states solved down to 3.7 angstrom and the other one down to 4 angstrom. Particularly in this case, you see that the cytosolic domain, so this is this A, N, and P domain of the P-type ATPase subunit, is not so well resolved. So if you play with the threshold, you see it's of course still there, but the resolution is hampered. So we also think that we might have here an additional dynamic. So this might perhaps even be able to separate into even more subtle conformation, different conformations. While state 2, it's actually better. I mean, it's a little bit lower resolution overall, but here we had a more confined conformation. You see also from the helix density that the building was straightforward, and this is of course where the X-ray structure became very handy. I mean, it doesn't mean that you get scooped, it actually just helps you, because with cryo-EM, if you work with these type of proteins, well, if you want to stay realistic, you normally do not get the resolution that allows you to very confidently build a de novo model. So if you actually have any structural information, you can just benefit from it. And we did. So I think we really did. So we built a model into it. So we have 2 populations. What are we looking at? So these are again, with the same color code to help you, we have these 2 states. And you can already, well, I already see the transmembrane domain doesn't look much different, but here a lot happened, and this is exactly what you expect. Also, if you remember that one video right at the beginning, P-type ATPase, they really move quite a lot around here in this region. And the way how you assign this E1 and E2 nomenclature really comes down to the relative position of these 3 subunits to each other. And what is more important to look at is the relative distance of this so-called TGIS motif. So this is actually the loop which will dephosphorylate the phosphorylation site, which you find in this blue subunit. So if they are well separated, this is then an E1 state, and if they are close together, so already about to dephosphorylate, then you know that you have an E2 state. So this is straightforward from a structural point of view. There's no ambiguity that we can say that this is an E1 and this is an E2 state. Of course, at this resolution, particularly the lower resolution we have in the cytoplasmic domain, doesn't allow really to define if we have those inhibitors bound, because for this you would really need much higher resolution. Nevertheless, with our collaborators, we performed some EPR studies, so electron paramagnetic resonance studies. And what you can do here is by labeling with the spin labels your protein, you can then measure the distance, the relative distance of these spin labels to each other. And I mean, this is now again the 2 states that we have, so we can calculate what is the distance that we would predict by spin labeling these sites. And then we measured EPR on different sets of inhibitor cocktails. So, and of course, for example, the wild type without any inhibitor, we see a large distribution of distances, which makes actually sense, because you would expect that in the wild type it actually just moves around the whole time, so it can go through all the states. And to make a little bit of a long story short, and I want to go through all these peaks, but by analyzing a little bit more carefully all these peaks, we are confident in saying that state 1 is most certainly an E1 phosphorylated state with AMP-PCP bound. So this was one of the inhibitors that we had. And the second population that we have is then the E2, probably also phosphorylated AMP-PCP and aluminum fluoride bound. So, but for the rest of the story, this actually doesn't matter so much. So don't worry. So what is new? What can we learn? So now, together with the X-ray structure that's published, we have 3 structures of the protein, 2 in E1 state. So this I forgot to put it here. This is also an E1 state. and one in the E2 state. And you can already, again, see by eyes probably that nothing seems to change much here, but quite a lot changes here. So we really have 3 states, captured 3 states of this complex. So the big question, does KDPA, so this is the channel subunit, open? So if you superimpose all of the 3 available structures, and remember we have 2 E1 states and 1 E2 state, so we are really actually having both sides of the transport cycle. So if, now it becomes very intuitive, if KDPA would open, you would really expect to see it now that something changes. And what is remarkable, if you superimpose all 3 structures, are virtually identical. So this kept us thinking, and I mean, how the hell does it then work? Because I mean, for decades, people believed that this at a certain point will just open and leave the potassium go through. And also, if we try to calculate any of these tunnels, so these accessibility cavities, we say, so here for state 1 and state 2, we were not able to ever find anything opening here to the intracellular, which is what was predicted. And this is just the example of another homologue of this type of family, so this SK2T family, so this potassium transporters, which have a very similar fold to this subunit here, also with this coupling helix, and there you actually see in an open state this helix, this loop moving away and opening a path for the ions to go through. But we really do not see it here in our structures. So what is happening here? So we then had a closer look into both structures. Again, more tunnels, so this becomes more and more intriguing. And what we see in state 1 is really a tunnel that goes from the extracellular side all the way through the selectivity filter and horizontally along this KDPA subunit down to the KDPB subunit. In state 2, we see a similar thing, but this extracellular tunnel is rather close. And let's just focus first on the upper ones. If you look at this, so this is now looking from the top. So here you have the channel 1, so this selectivity filter is here, and then you would go all the way down. So this is where this canonical ion binding site would be located in p-type ATPases. And this is shortened in the state 2. And this is very similar to what our other colleagues had found in their X-ray structure, where they also saw this tunnel, which they explained to be where water just flows through and senses a change in charges. So these are what we call our entry tunnels. And if you look at it, so here it ends here, there's nothing happening. But in state 2, we found another tunnel down here. So if you have just a closer look into that, so again, so this ends at the canonical binding site. And what we see happening here is that there are a large rearrangement of these subunits and also the helices in the transmembrane domain moving away and really opening a path. And what you also have here is this disruption of the salt bridge. And if you still remember, also from the introduction, this is exactly also what the authors had suggested, that actually the salt bridge, the moment it gets disrupted, or with this salt bridge, you could actually pull on this coupling helix. But nothing changes. I mean, this coupling, so we have this disrupted, but the helix is still where it is. But we have here suddenly a cavity which is open to the intracellular side and actually could probably go all the way up down to the canonical binding site. So taking this together, what we then suggest is that the state we captured with in this structure is an E1 outward open state of the protein, while in state 2 we have an E2 state, which is inward open. Further, we had a more closer look into the tunnels, and we realized that we were able to identify 3 unassigned densities for this structure, and in this structure we were only able to find 1. And I mean, at this point, of course, with cryo-EM, it's impossible to prove that this is potassium ions, but this is what we very strongly suggest, because this is a potassium transporting unit. It goes all the way from the selectivity filter down to here, and it actually makes quite some sense. So we actually believe that we see the ions moving through it. So what we propose now is, this is now just a little bit of an animation. So this would be the crystal structure, which we believe to be at the beginning. There was just 1 potassium bound. Let me just potassium bound. Then this is our E1 state, where we had 3 potassium bound, and then you have the further helix rearrangements to the other state that we have. And if you now just look closer to it again, remember this would be the canonical binding site known in p-type ATPases, and what we have in the first state is really a tunnel which goes all the way from the selectivity filter down to here. And what we believe that happens is that this is closed, so at a certain point it becomes closed, so the potassium can go through here, and then we have here this helix rearrangement. So once the potassium is here, it senses it, induces this helix rearrangement, the salt bridge is broken up, and we actually have then here an inward open tunnel where the ions, with the one potassium ion that was trapped here, can then be released. So the whole point of how you believe that p-type ATPases work is that this is normally a very high affinity site once it's open to the outside, so it attracts the potassium to go. But once this conformational changes are induced, you of course change the chemical environment there, which will then lower will just disrupt the affinity. And once you have then an open cavity, the potassium ions will then just go into the cytoplasm. So again, just to also make it again, maybe going one further. So just again as a comparison, what has been proposed until now and what we actually propose now. So at the beginning, it was always believed that you have this spatial separation of a transporting unit and an energizing unit, and they just somehow are coupled to each other, but do not participate both directly in the transport. So what was proposed is that you have this tunnel, which others already found, so that potassium binds in the selectivity filter. This difference in charge is sensed by this proton wire, induces conformational changes, and these conformational changes pull on this loop. Voila, we have the channel open, and somehow this thing then just tips over and closes the gap. So what we very strongly believe is happening now is that we don't have this case, but we actually, that the KDPB, so this subunit, still is directly involved in potassium. And the only thing that happened is that it just recruited a channel for different reasons, but still does most of its work itself. So that potassiums do not bind from the outside directly to the subunit, but they go first via this channel subunit from the selectivity filter, then horizontal through this tunnel, end up in the canonical binding site, and from here on we would have a rather more classical transport mechanism as found in other p-type ATPases, so that the conformational change induced here open up another tunnel, so you really have the alternating axis mechanism, and potassium is released. And as you can see, we actually do not need anything here for this other subunit to happen, and this is again also well, again, not a proof, but it's fairly confirmed, because again, in all available structures, it doesn't move. It's virtually identical. So it's hard to believe that this has the tendency to somehow move and act like a lid. So what could be the function of it? Well, we don't know, but we postulate, because if you look again at classical p-type ATPases, some of them have an additional subunit, which to some extent looks similar, at least from the position of it, to what we have here. And what they are presumably there for is to increase the affinity of whatever ion is supposed to bind to the subunit. So perhaps this is exactly also what's happening here, because it's located where the potassium ions enter this whole complex. So altogether, what we propose is a really completely new transport mechanism, and we have a true chimera. So this is really for the first time, to my knowledge, where you really have two different groups, which are normally classified into different groups, really coming together and working together. So what we believe that happens is that, so this would be with the three available structures somehow in the cycle. Maybe you will always catch some sort of intermediate. And what we believe that happens, I mean, this is more of a repetition now, so that potassium binds through at the subunit, and the whole reason why it's recruited, so why this p-type ATPase recruited this additional subunit, is really just to increase the selectivity and affinity. So potassium binds here, go through the tunnel, induces conformational changes, this closes, this opens, it's released, and a new cycle can begin. And this actually makes a lot of sense, because this mechanism also explains how it can pump so efficiently against such a big gradient and still retain this an incredible high selectivity and a very high affinity, which is required for of course this. So we believe that we really are able to, well, strongly propose this new hybrid transport mechanism. What I like about it, it blurs this mechanistic concept and this almost dogmatized categorization of different mechanisms. And what I also like about it, it's a perfect example which shows how conserved protein architectures can actually adopt to fulfill other functions. Because if you remember, I mean, why should nature come up with something, increased affinity of this subunit, because it's required for its survival, if it could just hijack already protein architecture, which has evolved to obtain that high selectivity and affinity. So we really think that we trap something which obviously happens rather rarely in nature, and we like it. Okay, so I had also promised to just give a few slides also on our use of the 200 kV machine in our house. So everything I showed you was done in-house on our Talos Arctica, and we have actually been quite successful, so it made me really happy. So we have been doing a couple of collaborations, and this has been all in-house. And just very briefly want to mention it just to show also the power of the machine. So one of the other projects is human ACT2, so it's an amino acid transporter, and we were able to solve it to 3.8 angstrom. And it's also a fairly small protein, well, at least it's a trimer, so that facilitates already image processing. But what I found very characteristic, and I was very happy actually that it works so well, is if you just look at the declassifications, we have here a very peculiar case where the protein is fully embedded in the membrane. And for everyone that does single particle knows that this can really be a problem during image processing. So this is in detergent, so it's not a nanodisc. So probably nanodiscs might actually become a problem. But we were really happy to see that we are not hampered by that limitation. Another project that I mentioned also right at the beginning is also this TMM60 family, which encompasses channels and scramblers, so it's also this dual functionality. Here there are also 100 cells up to 200 kilodalton. These are also human-like, so in HEC cells expressed proteins or in yeast, and here we have already obtained several structures, with some of them really going down to 3.1. So this is also possible. So this is not a proof of concept. I mean, Gabe Lender from Scripps has shown a couple of times as a proof of principle that you can obtain very high resolution with these machines, but this is of course with samples that behave perfectly. So these are rather challenging proteins, membrane proteins, and we still are able to go down to 3.1, and I'm pretty sure that at this stage we were more limited by the protein itself and not yet necessarily by the microscope. But this is clearly where it becomes more difficult to face it. I have one thing I actually also like about this this family is that also resolving the nanodiscs. So this is where we actually see one of my favorite take-home messages of this story is where we also see a bending of the membrane, and I think we also believe that this is functionally important for the protein. So why do we like, or what don't we like about our Talos Arctica? So this is Gert. Gert is also in the audience. So Gert is the brain of this microscope and our facility, I would say, and we are still very happy with it. So what are the advantages of it? Well, first of all, obviously it's cheaper than a Titan price. So if it comes down to buying a microscope and convincing people to provide you money, if you need 2 or 3 million less, well, you might get the money. If you need it more, you will not get it. So this is, of course, often the main reason for it. Another advantage is it has the same autoloader mechanism, so this makes it compatible with the Titan Prize and other workflows of Thermo Fisher or FEI. But probably what is really a big pro, and this is of course only beneficial for, or mainly beneficial for single particle, is at 200 kV compared to 300 kV, you have a higher contrast. And we really believe, so this is clearly good for single particle, and also certainly very beneficial if you work with small proteins, which is what we are working with, and also membrane proteins where you have the problem with the micelle or lipid nanodiscs and so on. So this is, we think it's where we're actually having a benefit out of it. I'm not sure if this number is correct, because it's sometimes really hard to keep track of what's happening. I know for the Titan, the current breakthrough is 1.5. For the Talos Arctica, I think it's 2.6. But And the important take-home message here is, of course, with the tightness, always better, and you will always obtain higher resolution. But at least with the proteins that I'm working with, membrane proteins, so my targets are actually normally the resolution limiting factor and not the microscope itself. So this is why I'm often not so worried about, because I'm realistically anyway not aiming for this resolution. That would be awesome. So what are the clear disadvantages? Well, first of all, it has a slightly worse vacuum, so this can become a problem if you talk about ice contamination and leopard skin, and also the amount of time you can actually keep a grid in your microscope. Well, here we have to admit that it looked much worse at the beginning. Now we found a way around it, and it's as simple as it was with the suggestion of just doing a cryocycle every 3 or 4 weeks, and this really helped. So I think Gert also, you thought it's rather counterintuitive, but this really helps. So this might also be something to consider. So we could optimize this a bit, but it's still worse than in the Titan Cryos. Of course, if you're working with the 200 kV, you have a limitation in thickness, because you just, until which thickness it is still transparent for this voltage, and this is of course a bad thing for tomography, not necessarily for single particle. There you anyway aim for very, very thin samples. And probably one of the biggest problems of, well, the setup of, it's not because it's a 200 kV, but it's just the setup of the Thales Arctica, or actually all 200 kV machines out there, is you don't have a third condenser lens. And what this means is that you don't have a range, a beam size range, where you have parallel illumination, but you really just have, for given settings, one specific combination of settings where you will have parallel illumination. And I don't know if you noticed, I put this actually as rather yellow because this is a disadvantage, but from a practical point of view, there's again something good you can get out of it. And the fact is, well, we spent quite some time at the beginning when we installed the microscope at Groningen to really check every little thing of the microscope, find the best settings, and I mean, I'm working with small proteins, so at a certain point you have your best settings. Once you found them, we're not changing basically anything. And if you don't change so much, it actually makes your life also easier. So it's a disadvantage which you can turn into an advantage. So I think one of the positive things of here is, I think this also translates into the stability of our microscope, which has comparatively been not that often down, because you just don't tweak around so much. And also from a practical point of view, for the people using it, it also becomes more straightforward. I mean, they don't have to think about everything, which I don't like so much, because I think everyone should always very carefully think about why I'm doing it this and this way. But still, from a practical point of view, it's not that you always have to check what beam size do I have, which electron exposure will this be, and what does this implement like, all these kind of things. So we have one defined settings, which we use the whole time, and this is also convenient. Another problem that arises from this not having a parallel beam range is you cannot adjust the size of your beam. So for whatever combination of settings you have, there's just one beam size, in which way you will have parallel illumination. If you change that, you are out of range. So, and in our case, for our settings, it's quite big. So I think it's at 130 kx, it's 1.7, 1.8 micrometers big, so which is exactly the size of the holes that we use. So luckily, we can still acquire one image per hole, so we don't have to skip holes. But we cannot acquire more than one image per hole. And this is something that you can very nicely set up at the Titan Cryos, which speeds up tremendously data acquisition. So this is a clear disadvantage from a more practical issue. And there are also some state drift issues. So this is something I'm just going to present this now. So this is now something that FEI is also aware of, and they are actually trying to solve the problem. And this is where I also would suggest every time you get a new microscope, really test it to its bone. And this is something that Gert really did at the beginning, was really checking all kind of things. And if you look at the stage, this is just an overview. If you now move it in the Y direction, in the dashed line is what is the specification, and you see that it behaves beautifully. But if you move it in the X direction, this is not the same. Of course, there are differences in where of the regions you're moving it. You actually have then a severe stage drift appearing, which is way above or way below the actual specification. So it's worse than you would want, and it's very bad because you see it would actually take you, you see it here, more than 10 minutes to still not having gone down to where you would want to have it. So this is something that really limits it. So it's not that easy as depicted here. So it's not that you have just one bad region and the rest is perfect. So it's not that simple. It's well, there is a region which is a little bit worse, but it's really about from where you move in which direction the stage. So there are regions that cannot be used or should not be used, and in general avoiding drift so that the data that you collect is of high quality can cause or will cause extreme delays. So just summarizing this, so basically the problem, so also the stage drift might have an implication of the camera you want to use. I put this in brackets because, I mean, if you have a stage drift, this is something you will not be able to correct for with motion correction, right? So because within everything that moved within the frame you collected, this will not be able to be corrected. So if you now think about Falcon camera, which has a very long exposure time, this would become a serious problem. But I put this in brackets because the last time we spoke to Thermo Fisher, apparently the Falcon 3, they have implemented, they are tackling this problem. So they have, well, I call it the software implemented, it's implemented in the hardware, where they really integrate in between the frames so that they actually can correct for this stage drift. So it seems to be fine, but all these implications you have to consider. But taking the problem with the beam size and in general the stage drift, where you just have to wait sometimes a little bit longer, comes up with a actually severe problem. And to collect a high quality data set at the Thales Arctica will take 2, if actually not 4 times longer than if you would do it at a Titan price. And if you now take this one and my first message, you will see that from an economical point of view, a Titan price in the long term would still be cheaper, but of course this is unfortunately not how you could argue. Nevertheless, the Thales Arctica is a wonderful machine where you can do quite a lot with it, and we really try to make the best out of it. How do we do this? Well, first of all, by really knowing the problems that you have, so communicate with it, test it. It's like a marriage, so if you know what the problem is, you can deal with it, and at a certain point you accept it and even start liking it. So what do we do? So this is our setup. So those 3 screens are the typical, well, this is how you operate the microscope, this is how you operate the camera. So we have a K2 with an energy filter, and what we have on site is also a powerful GPU machine. And what we really do to make it as efficient as possible is we have a way, so this is now fully Gert's contribution. We have an ice thickness measurement, and the way how you do this is it's pretty simple. So first of all, of course, from the scattering of your object aperture, you have already would already have a difference in your incident beam to the scattered beam. So when it passes the specimen, but what is really gives you a much better signal is if you have an energy filter, you can make use of it, because you can then simply compare the incident beam without an energy filter and with an energy filter, and this then from this on you can then calculate the thickness of your specimen. This is really what we do in-house. This is a script that Gert wrote for digital micrograph. So we calculate the thickness from the ratio of the unfiltered and filtered, and he even wrote it that it's color-coded, so we can then set here the exact range of what we know it's optimal. Also, this might vary from project to project, and then we have it nicely colored. So if it's not colored, it's too thick. If it's red, it's too thin. If it's green, it's optimal. And if you have this knowledge, I mean, we still use EPU. I mean, it's to implement serial M is on our list, but we never found time to do it, so we still use EPU. And everyone who's familiar with it, you have this so-called ice filter in EPU. So this is where you can define which holes he actually picks when you do the automated data collection. So what you then just need to do is really take the knowledge of, well, look at this image and then just adjust the ice filter in EPU so that you really just end up picking those holes which have the thickness that you want. And this might look like a bad example, but it actually shows you that if you would have done this manually or automatically by the program, you probably would have picked all of these holes. And if you look at what the holes are, the quantity of the holes, that probably will give you the quality and the resolution that you actually aim for. Well, if you would have collected all of them, at least 3/4 of what you had collected, you probably would trash or would simply not contribute to your higher resolution. So this is how we try to make it, the data collection, as efficient as possible, also to compensate for the fact that we just in general need longer. My contribution to the setup, so I always like to say I lived so many years in Germany. One thing that I very proudly adopted is trying to be efficient as possible. So the way how we try to do this is we have this ice thickness measurement, we have already our numbers and we kind of know it, but nevertheless we always set it up, start data collection and check on the fly how the images look like. And this is where focus comes in place. So there are several different programs you can use to do this. I like Focus, it's from Henning Stahlberg, because we just liked also the whole interface. So what happens here is that we feed this program, so this is then on this GPU machine, and what we do here is this machine automatically feeds itself with the new images that are coming in, and it just does the motion correction, it does a CTF estimation, and gives you this wonderful, nice overview of it, where you then also can sort it according to the defocus, to the CTF resolution estimation, to the counts, which for us is also basically we can translate into the thickness and the drift and even iciness. So you can even select for all these kind of things. Then on the side you really literally see your image, so you can see if there is dirt, if you see your particles or not. You see the CTF, you see the CTF estimation and how bad the drift was. So and the nice thing about this table, so first of all what we do is we really record a couple of images according to the ice filter we set up and just check if this is actually making sense, if this is giving us the quality of images we want. So one of the striking ones, of course, always to look at the CTF estimation, the resolution estimation. If it's not, we simply go back and adjust it again, so that we really have and this is really within a minute you have your answer. So it's what we do, and also another thing which just makes it efficient in the whole cycle is once you're done with your data collection, you can use this table to simply sort according to it. So, and once you sort it, imagine you say, well, everything with the CTF resolution estimation worse than 5 angstrom or 4 angstrom, you just want to trash, you just click, select all of them, and with one click you have trashed them. So, and with this, we also make sure that we only export what we really want to process. This is also just from a data management point of view, saves you time. And I remember still during my postdoc, I did all of this by myself at the computer, opening every single image myself. Still, this reduces me almost a week of work at the computer doing it. And taking all of this together, we now, for most of the projects, we really managed to, in best cases, to have just 10 or 20% the most, which we actually end up trashing. So that the time that we use at the microscope is really very efficiently used. Of course, this doesn't apply to all of them. Sometimes you are lazy and just, well, let it collect, or you know it's not that good data set, but you still want to look at it. Yeah, so, and this brings me already to the thank you slide. So I talked about the KDPF-ABC project. These are our collaborators from Frankfurt. So Inga Henold is, where is she? Oh, here it is. She is also assistant professor. And the main contributors were then Charlotte and Dora doing the EPR, and Igor also with the structure building, everything. This is my group in Groningen. So the work that I showed you was mainly done by Lisa, so she's here, and Gert, of course, who has helped me quite a lot and everything. This is how I rather like to depict my group. So I would say we have fun. And, okay, of course, very important also, all the funding. Without, I could not pay all my people. And, well, just as a last anecdote, because I think you never saw this movie, so when the boss is away, I actually get this type of videos of what people do with the microscope. You remember I have a powerful GPU machine next to it? So this is what they do when I'm at a conference. They also have fun without me. Yeah, that's it.
Thank you for beautiful structures and explanation of the workflow. It's very nice.
Sure.
Questions?
Hey, it was amazing, thank you. My knowledge of transport proteins and biology isn't great, so this might be a stupid question. But I understood that the orange one with the diamond inside, classical ATV, usually opens at the top and then opens at the bottom. And you showed really nicely how it kind of comes in from the side. What's stopping it opening from the top? Because now you don't have that purple lid there from before.
Yeah, so what stops it, well, yeah, so you're completely right, normally it would open like this. The reason why we think it doesn't open to the bottom is you also just have the other subunit just next to it. So this is probably already closing it up as well. Then the whole transmembrane domain is also a minimal transmembrane domain. So normally you have 10 helices, here we only have 7.
Okay.
So you also have some missing that would also help to open it up.
Right.
But mostly we think it's the interface, so that you basically just have this other subunit next to it, so it can't even open to the outside, but rather opens just slightly, so that you have horizontally really at the center of the membrane then this tunnel going through.
Okay, thanks.
This is, well, how we...
Practical question. You said you have more ice content issues, so how long can you realistically imagine?
Yeah, so right at the beginning we were actually very worried. So at the beginning our leopard skin tests told us this the most 48 hours, and then you should actually trash them. But this of course also depends what you're looking at. We are dealing with small particles. So if you start having leopard skin, so everyone is aware what that is, so this graininess, so it's when the eyes start falling on top of your sample and you have this graininess. I mean, if you're working with a ribosome, a little bit of grain will not hamper the quality of your data. But if you're working with small proteins, that grain starts being the size of your protein, then you of course get into big trouble. So at the beginning, our take-home message was no longer than 2 days, which is of course was very bad because just collecting data took so much longer. So we had to trash at the beginning very good grids, but now it's much better. So now I think we even do 4 days, right? But then after 4 days we normally stop. Yeah, but 4 days I would say is still okay. And it's really just a cryocycle, so it's actually, it still amazes me. Yeah. So and it actually gets better all
So
the
with
time.
cryocycle, you mean you should not cryocycle that much?
Yes. Yeah, so what we do now regularly, if possible, weekly, just the autoloader, if possible. I mean, if we have data collection, then we can also skip that. So the autoloader I would still regularly cryocycle. The column, we only cryocycle every 3 or 4 weeks. Yeah. And it really works wonders.
Also got a question with the workflow. I was wondering, it's all very nicely set up, very perfect. How sensitive is it to a service engineer coming from FEI and getting back to that optimal setting? So every other day is the setting exactly the same, because there might be more than one user. Where it's very, very stable. It's back to the original setting.
So do you mean now the whole...
The whole setup, measuring of the ice thickness,
So-
all the little
so-
things.
All those little things
Yes, the
are
whole
after
workflow.
the microscope. So there's nothing that they do with the... So this is really separate. So it's even in a physically different computer even. So we of course just in between make use of the programs, but this is really separate. So there's nothing. And with the settings, I mean, it's the same as with any other microscope. I mean, you can of course, if you want, lower and use other settings. I mean, you still have then to know exactly which one. So it takes perhaps some time to find the right parallel illumination. I mean, this is all the things you have to do yourself. But if your FEI engineer comes, it's fine. I mean, we normally have our effect registers, which brings you very close to what we had before. And then, so normally whatever you do, and has been very stable, whatever you do, it's go back to the last settings, which were saved, and you just do the high-end alignments, and it's actually only fine. Yeah.
Right.
But everything I showed you this after is really literally after. So it has we did not tweak or change anything. The unit would not even be allowed to do that.
I could imagine that there are certain hysteresis steps somehow that if you go from one elimination to another, that might be
Oh yeah,
different.
this we avoid. So this was actually a problem with the ice thickness measurement. Before we did not use the trick with the energy filter on, energy filter out, because there you have the problem. Normally you would have to go to a magnification that is too low so that you go out of the LM mode. But now we actually, I mean, you can still work around it. So now we use it. We have the smallest.
If there are slightly changes in your centric height, that certain shifts, calibrations are a little bit off, that kind of things. I'm not sure if that's true, but perhaps not.
Nothing out of usual. In that sense. I mean, the stage, if you tilt of the Thales Arctica, is also a little bit worse. So if you, I don't know how much this would influence tomography. So you can actually, if you do the wobble, so the alpha wobbling, you actually see that it doesn't go like this. It goes and then swaps a bit. I have trouble in actually. So it's not like this, but it goes like a little bit like you can actually see it. So, but I don't. Yeah.
And why is this?
It's also the stage.
Yeah, there's a bit more of looping also of the stage.
Yeah.
If you alpha wobble it, it also moves a bit in the direction of the holder more than a good trios stage would do.
So it really turns out the stage is
So it's a
physically
feature
different.
which you can see it from different angles then, not only single will be different other angles. It's a large angle
You see?
filtering.
Yeah, the
Yes.
same optimist as I.
Monza will love this. I
Have
understand.
you done tomography? No. Have you tried for tomography or not?
Not really, no.
But isn't the Arctica stage still the ultra stable stage that now?
Supposed to be the ultra stable stage.
That's what FBI wants to want you to believe.
Yeah, no. So
It's definitely not.
so the whole thing also with the stage.
So
This
what
is
is then the ultra stable stage that, like for example, Francis has?
It's this stage.
What's the
It's this
Arctica
stage.
stage?
It's this stage. It's no different.
Right, let's have a beer and continue the discussion
Sure.
over beers. Right there. All right, thanks everyone again, and thanks.